Welcome to this explainer. Today, we're diving straight into a microscopic detective story to uncover how the brain's own cellular protectors can actually become its worst enemies in neurodegenerative diseases. We usually think of conditions like Alzheimer's or Parkinson's simply as diseases of dying neurons. But modern pathobiology reveals something far more sinister going on. It's really a betrayal from within. Just to grasp the sheer massive scale of what we're talking about here, consider this staggering estimate from the World Health Organization. Nearly 1 in 3 individuals globally are affected by neurologic disorders. 1 in 3. That makes them the leading cause of disability and the second leading cause of death worldwide. And with our aging population, these numbers are expected to literally double by 2050. So the urgency to figure out what is actually happening inside the brain, it's really never been greater. Here is our roadmap for the investigation today. We're going to cover the burden of brain disease, microglia as the brain's gardeners, toxic astrocytes, protein aggregation and prions, genetic drivers of disease, and finally, stem cells and disease modeling. Section 1. The burden of brain disease, a global crisis in neurology. Okay, as we just discussed, the global burden is absolutely immense. But to stop this crisis, we have to zoom all the way in to the cellular level. We need to take a really close look at the brain's intrinsic immune system. Section 2. Microglia, the brain's gardeners, and our very first cellular suspect. Now, what's really fascinating about our brain's innate immune system is the dual nature of these microglia. They're a highly specialized macrophage population that actually comes from a primitive cell group in the embryonic yolk sac. They migrate right into the nervous system before the blood-brain barrier is even fully formed, which makes them lifelong residents of your brain. In a perfectly healthy brain, these microglia act as the ultimate gardeners. They're constantly maintaining homeostasis by clearing out cellular debris, taking up loose proteins, and they even release brain-derived neurotrophic factor, or BDNF, which actually promotes neuronal survival. They're even out there helping to sculpt neural circuits by precisely pruning away weak excitatory synapses. But here's where the betrayal begins. When they get provoked by toxic proteins or an injury, these helpful gardeners turn into deforesters. They literally change shape, shifting from a resting ramified state, where their long branches are just constantly surveying the environment, into an activated amoeboid shape. In this state, they become completely hyperresponsive. So instead of just clearing dead debris using complement proteins like C1q and the CR3 receptor, they go rogue. They start pathologically targeting and destroying perfectly healthy neuronal synapses. Section 3. Astrocytes, protectors turn toxic, the radicalized accomplice. Our next suspects in this microscopic crime scene are the astrocytes. For a long time, astrocytes were basically thought to just be simple structural glue holding the brain together, but we now know they are incredibly complex, dynamic cells that make up over half of all the cells in your central nervous system. And here is the crucial point. Those activated microglia, they don't act alone. Normally, astrocytes are maintaining the blood-brain barrier, vascular tone, and supporting healthy synaptic pruning using something called the MEG10 receptor. But those activated microglia start secreting specific inflammatory cytokines, specifically IL-1A, TNF, and that complement protein C1q we just talked about. These signals effectively radicalize these essential astrocytes into what is known as a toxic A1 state. And once they transform, they completely abandon their protective duties and actually start killing off neurons and mature oligodendrocytes. Section 4. Protein aggregation and prions, the microscopic weapons. All right, we've met the compromised cells, the microglia and the astrocytes. So let's go even deeper. Let's move to the molecular level and examine the actual weapons causing this damage and setting off this whole inflammatory cascade in the first place: misfolded proteins. To really get this, we have to understand the prion. The prion concept can be a little tricky, but essentially it's a corrupted protein, one that is entirely devoid of DNA or RNA that adopts an alternative beta sheet-rich shape. And by doing this, it gains a pretty terrifying ability. It undergoes self-propagation. It basically acts like a localized contagion right inside your brain tissue. Let's walk through exactly how this happens step by step. Step 1. You have a normal precursor protein just doing its normal job. Step 2. A rogue, misfolded prion comes along and binds to that healthy protein. Step 3. Through a process called self-templating, the prion forces the normal protein to adopt its toxic beta sheet shape. It's like a molecular domino effect, which leads to Step 4, an exponential spread of dysfunction,
with these misfolded proteins assembling into massive amyloid fibrils that just choke up the cellular machinery.
For the longest time, we thought these diseases were totally unrelated, but when we map these specific molecular weapons to their respective neurodegenerative diseases, the underlying prion-like mechanism is strikingly similar across the board. In Alzheimer's, the self-propagating proteins are A-beta and tau. In Parkinson's and Lewy body dementia, it's alpha-synuclein. And in Creutzfeldt-Jakob disease, it's the PRPSC prion. The specific protein changes, sure, but that self-templating mechanism of destruction, it remains exactly the same.
Section 5. Genetic drivers of disease. Unlocking the brain's doors. Okay, let's look at the genetic mutations that act as accomplices here. You might be wondering, why do some people's brains succumb to these protein weapons while others don't? Well, the answer often lies right in our DNA, which can leave the brain vulnerable to these cellular betrayals.
These specific genes basically dictate your risk, and they tie right back to our cellular suspects. Take the ApoE4 allele, for example. It's expressed primarily in astrocytes, and inheriting 2 copies of this drastically reduces the brain's ability to clear amyloid, spiking Alzheimer's risk by 10 to 15 times. Then there's TREM2, which is a vital microglial receptor. If a mutation causes a loss of function there, the microglial cleanup crew is basically crippled, which quadruples Alzheimer's risk. And mutations in the GBA1 gene impair the lysosome's ability to break down alpha-synuclein. Plus, we can't forget about defects in retromer trafficking. That's the cellular shipping system that sorts and recycles membrane proteins. When that system fails, it traps these toxic aggregates inside the cell, accelerating the destruction. Together, these specific mutations are directly responsible for a significant chunk of cases, like the 5% of all Parkinson's cases driven entirely by GBA1 issues.
Section 6. Stem cells and disease modeling. Fighting back in the lab. So the picture looks pretty grim, right? But let's look at how modern science is modeling this microscopic crime scene to fight back. We aren't just observing this destruction anymore. Scientists are literally recreating it in the lab to figure out exactly how to stop it.
This is where things get absolutely mind-blowing. By taking a patient's own skin or blood cells, scientists can create induced pluripotent stem cells, or iPSCs. This means researchers can grow a specific patient's exact neurons in a dish. They can even build 3D cerebral organoids, which are basically mini-brains. With these, researchers can watch the toxic prions accumulate, watch the microglia get activated, and test entirely new therapies on a patient's actual unique biology, all in real time.
I'm going to leave you with this final thought today. As this explainer has shown, we now intimately understand the cellular suspects. We know about the microglial gardeners turning into deforesters, the radicalized astrocytes, and those molecular prion weapons. If we can now perfectly model this exact betrayal in a laboratory dish, how long until we can stop it inside the human brain for good? Thanks so much for exploring this fascinating pathobiology with me, and keep questioning what you think you know about the brain.